This video is membrane structure, membrane proteins 1. I want to start out by emphasizing what exactly we mean by the membrane, and I'll do that on this slide. So basically, membrane proteins, these are any protein that's associated with the lipid bilayer of the cell membrane. So let's talk about what exactly the membrane is. Certainly you've heard a lot about these in many biology courses. And basically, I'm going to start off with specifically pointing out our membrane structure is beyond phospholipids. And so basically, if we draw the membrane, we've got our phospholipids here. Also have membrane proteins like this one. And we could also have a membrane protein that has some sugar molecules attached to it. And basically the point here is to say that this whole thing is the membrane, and therefore this includes phospholipids, membrane proteins, and glycoproteins, to name a few. There's some other miscellaneous items that we'll run into at other points. So you might not know what a glycoprotein is, so I want to talk about that. For example, the glycoprotein is what's shown here, and it's kind of like what it sounds like. If you break down glycoprotein, glyco sounds a lot like sugar or glucose, and in particular, this is some sort of saccharide chain. And in particular, the definition from the book is an oligosaccharide chain, which means that there's a number of saccharides that are covalently bonded to one another. And so this oligosaccharide chain, which I drew kind of like this, which is not my best drawing, but here I'm drawing some maybe glucose sugars that are all bonded together. They are also covalently bonded to a protein. And so together that's a glycoprotein. And so if we're going to write out that definition, this is a protein with one or more covalently linked oligosaccharide chains. And these are particularly important in terms of cellular identification. Glycoproteins are commonly found on proteins that are exposed on the outer side or outer layer of the cell membrane. In that case, meaning this would be the inside of the cell and this would be the outside of the cell. Glycoproteins can also be secreted proteins. So a protein that's secreted may have these additional covalent linkages. And so it's kind of a hybrid molecule from what we've talked about so far. We're going to learn more about the other types of membrane proteins very soon, but here we see a membrane protein. And membrane proteins are incredibly important. They carry out most of the functions that happen as a result of the cell membrane. Whereas phospholipids provide a barrier, since we have the hydrophobic portions, the fatty acid tails, and the hydrophilic portions. But the phospholipids pretty much have the same role for any membrane, which is to primarily keep most molecules out. So let's talk about some of the membrane functions. So in total, across all these different components which make up the membrane, there are several different roles. And these things are really key for defining what a cell is and what a cell does. These particular components will and can vary across different cell types or for given conditions for a particular cell. But one thing is shared for all cells, and that is that they have a membrane. And basically, here's a couple of the roles that membranes have. So they can form a barrier, and they do form a barrier. They also allow the cell to form compartments, especially when we talk about organelles. A key defining factor for an organelle, like mitochondria or chloroplast, is that it has a membrane, at least one membrane, around it. Membranes are also a meeting place or shared localization spot for certain enzymes or other proteins that are in a shared biochemical pathway. And the membrane also includes molecules that are used as identification, which is actually a pretty common role for the oligosaccharides and the glycoprotein. Those are identification tabs that could identify a certain cell as self or non-self. Membranes also work as gatekeepers, allowing certain things to come in and other things to not come in, or vice versa, to go out. And basically, taken together, all of these components will serve these functions for the membrane. And basically, without a membrane, there's no cell and no life. Just to give you one example of some of the pathways that take place using membrane proteins or other membrane molecules, as well as the compartment, that includes both photosynthesis as well as chemiosmosis and cellular respiration. So those things you would have heard about in an introductory biology course. And basically, those biochemical pathways rely on not only the membrane proteins and other membrane localized molecules, but also the fact that certain things can build up, say, inside the membrane and then slowly be allowed out. A few other things to impress upon you: the makeup of the membrane is that the membrane is about 50% made up of proteins. So although we often just initially imagine the phospholipid bilayer, remember the membrane is both proteins plus the phospholipids. And proper functioning of proteins within the membrane is crucial for a variety of processes, including for neurons to transmit an electrical impulse. And also, there are membrane proteins which, if mutated, can cause deadly human diseases. And one example of those is cystic fibrosis. So those are just a couple examples about the importance of membranes. So neuronal signaling, and they can also be used for ion transport to maintain proper conditions within the cell. One example of a human condition which has improper ion transport is cystic fibrosis, which causes a buildup of mucus. It's kind of a long story, but it at the core has to do with improper ion transport, and then allows a very thick mucus to build up, which can lead to infections and potentially death. Okay, let's talk about 4 different roles or functions of membrane proteins in eukaryotic cells. So those are listed here and shown by this figure. This figure, by the way, is figure 11-19 in your book. And so those functions are: first, transporters and channels, and these are lumped together, though they have some subtle differences. And we'll talk about those shortly. Secondly, membrane proteins, some membrane proteins are anchors. Third, membrane proteins might have a role as a receptor. And fourth, membrane proteins could have an enzymatic role. So let's talk about each of these, basically starting over here with the transporters and channels. So this first example on the left, that shows us an example of a transporter, specifically the sodium-potassium pump would be one example. And basically the sodium-potassium pump, you should have heard about in introductory biology or maybe anatomy and physiology if you've taken it, but this uses ATP to change conformation and this allows sodium to exit the cell and potassium to come in. And so this is one difference between this transporter protein, which is again the sodium-potassium pump, and the big deal is that it changes shape. So that's one thing that makes it a transporter. And if you look at this little drawing up here, notice that there's this kind of open part here and then it's closed. And so basically that will sequentially open and close while it is working. On the other hand, this channel protein over here does not change shape while it's functioning. And an example of a channel protein would be a potassium leak channel. Both of these types of membrane proteins are used in neuronal cells. To transmit electrical information, specifically an action potential, and we'll hear more about them later. For now, it's important to note that they do have two separate roles, and the shape change is a big part of that. And just to kind of talk a little more about the channel, see how basically whatever this is can just kind of sneak through, and so we don't see any implication of one part being closed like it is for the transporter and open here. Okay, second role is anchors, and so one example of an anchor is integrin. And so that would be best depicted by this blue blob here in the middle of these three different sorts of anchors. What anchor proteins are attached to is the extracellular matrix, ECM, and again this is outside the cell, this is inside the cell. Your book seems to be pretty consistent with that. And so some anchor proteins are only bound to ECM, like this one here. Other anchor proteins are bound both to the ECM as well as the cytoskeleton. And in this case, if it were integrin, then this would be actin. And then you can see another sort of anchor protein here. It looks kind of like a weird mushroom, and it's only bound to actin. So integrin proteins, like their name sounds like, they integrate. In particular, they are going to link intracellular actin, intra meaning in, to extracellular matrix proteins. Third example of a function of a membrane protein is a receptor protein. And one example is PDGF receptor proteins. And so what that whole alphabet soup stands for is P is for platelet, D for derived growth factor for the GF, and this name kind of sounds a little bit vague, like what a growth factor is, but that's telling us some about what this particular molecule does. Basically, what this signaling molecule, if it says red dot here, that would be the PDGF factor. And then the receptor is the blue blob that binds the specific ligand. And basically the deal is when PDGF is present and binds PDGFR, its receptor, it stimulates mitosis. Which is a type of cellular growth, hence the growth factor. We will hear about a variety of different types of growth factors throughout the semester, and usually the first part of their name tells us what sort of cell type that they operate in. In this case, this has to do with platelets, which is one of the components of our blood, and in particular, these are used for scab formation. So if you received an injury, then part of your cells in the adjacent area would start to produce PDGF here, and then stimulate the other cells around it to go through mitosis and basically seal the wound. The last function of a membrane protein is shown on the far right, and that's enzymes. So we've talked about enzymes already, and so what we see here is there's kind of this like little star power thing going on, and so basically this enzyme is converting some molecule X into molecule Y. And the example that I'll give you, which we'll hear more about later, is adenylyl cyclase. So this is a membrane-bound enzyme. And its claim to fame is that it catalyzes the production of cyclic AMP, which we've heard about briefly already, little c, big AMP, not camp, but CAMP. And this particular enzyme's adenylyl cyclase catalyzes its production in response to extracellular signals. Our last topic for this video is to talk about association of membrane proteins with said membrane. And so basically there's several different ways that proteins can associate with a membrane or be localized. So we're going to talk through those. There are 4 different ways that are shown here, and you can see them all split out. So they can be transmembrane, associated with a monolayer. They could be lipid-linked or protein attached. And this is figure 1120 in your textbook. Okay, so let's talk a little bit about this drawing. So basically the lipid bilayer, here's the phospholipid tails and the hydrophilic heads. They'd be located like this. And so basically each of these proteins is sitting in a membrane in this drawing, which is shown here. And you can see cytosol, or inside the cell, is at the bottom of the figure, and this would be outside the cell up here. Okay, so transmembrane, this is when we have a protein that is localized through the membrane. Kind of through or across the whole membrane. These proteins are amphipathic, which means that they have both hydrophobic regions and hydrophilic regions. And basically what that means is that they're going to have some of their mass on each side of the membrane. So with this one protein here, this is a polypeptide chain, and you can see some part of it is outside, some part of it is within the membrane. This would be the hydrophobic portion, and then some part is inside the cell. And so basically both the outside and the inside portions that are shown here with the green squiggle, those are going to be hydrophilic. And there's a few different ways that this can happen. Basically, we see a little more complex structure here where this is a single polypeptide chain that zigzags in and out. So this is a longer polypeptide chain that has more hydrophobic regions, each shown by this curlicue here. And all actually are implied here to be, each of those is implied to be an alpha helix. This may also look familiar to you, this protein here with all the zigzags. So this is a beta barrel protein. And recall that what the portions of the protein which are located within the membrane need to be hydrophobic. So there need to be hydrophobic side chains that are associating with either the sides or the entire portion of the membrane. Okay, so that's the first type of way that a membrane protein can be located within the membrane. The second way is the monolayer associated alpha helix. And so that is this one right here. And so notice that this protein has this squiggly part. That's the alpha helix. And it is only located within 1/2 of the 2 layers of phospholipids. So it runs in this direction. So it runs along 1 layer instead of across the 2 layers like we saw over here with the transmembrane. And again, so our definition for these is that these are associated with just 1 layer of the phospholipid bilayer. And so in this case, the hydrophilic sides are going to be on the same side of the cell membrane. A third type of membrane-associated protein is a lipid-linked protein. And just like this sounds, these are going to have some extra link or covalent bond. In particular, lipid-linked proteins are covalently bonded to 1 or more lipids in the membrane. And so basically, these lipids are shown with this little red or kind of pink zigzag. So that's not that pink zigzag, which we see right here. That's not part of the protein. That is a different lipid. This is implying that it's a hydrophobic chain, not an amino acid chain, but a hydrophobic chain. And so that this particular protein in green, it has got a covalent bond right here. And so that is how it's going to be essentially kind of tethered to the cell membrane. These proteins that are lipid-linked are not amphipathic. In fact, they do not have large hydrophobic regions. Instead, they're just kind of glomming on through this covalent bond here. And you can also see there's a couple different ways this can happen. So this one is just kind of like bobbing along. This would be like if there was a boat that was tied up to a dock. So it's just kind of bob along. This one, this other protein here is more complicated chemical structure. It does have 2 links, 2 covalent bonds to these lipids that are located in the membrane. And then you can see that there's also some saccharide molecules here. So this would be a glycolipid. And it's even got phosphate groups in there too, and something else that's red. So this is a pretty interesting structure. Okay, and last example is the protein-attached membrane proteins. And here, the deal is that these proteins, which are shown in green, are bound indirectly to one side of the membrane. The important thing here is indirectly. And these indirect interactions are with other membrane proteins. So basically what that means is that we've got these protein attached membrane proteins. So the protein attached protein is this green one right here, or this one up here. Notice that neither of those proteins are even touching directly the membrane. However, they are touching another protein which is directly associated with the membrane. And so can you think about how that interaction would take place? So between two proteins, what sort of interactions have we talked about right now for them to bind to one another? So the answer is non-covalent interactions. So the protein attached are going to associate with other proteins through non-covalent interactions. So we also want to talk a little bit about a couple more vocabulary words, and that is integral membrane proteins. Basically, the first three I talked about, A, B, and C here, these are all integral membrane proteins. And the deal is that these are, as their name implies, somehow tightly integrated with the membrane, at least to some degree. And your book defines this as that you need a detergent to require these proteins from the membrane. So you need these, if in a lab, if you're trying to purify these proteins and maybe run a Western blot on them, this would require detergent treatment to remove them from the membrane, which is kind of a harsh treatment, and as we talked about before, dissolves the phospholipids from their membrane configuration. On the other hand, the protein attached is the only example we have here of a peripheral membrane protein. And so, as their name implies, they're on the periphery or edge of the membrane. And these require a more gentle treatment, so they only require kind of a gentle if you wanted to isolate these green proteins from the rest of the cell. And so basically whatever treatment would dissolve or kind of inhibit these interactions between the two proteins would be enough to pull this green protein or this green protein away from those parts.